Okay, about the anaphase. We know that one of the main checkpoints of the cell cycle is here between the metaphase and anaphase. So the start of the anaphase is well controlled. The passage between metaphase and anaphase is done by the key molecule APC, anaphase promoting complex. Okay. What APC does, well, it is doing several things, but it sets free the chromosomes. You know, there are cohesins still between the chromosomes, still keeping them together in the centromere. These cohesins are not broken because the enzyme, which is responsible of breaking the cohesin, separase, is kept inactive by another molecule, securin. Once the APC, anaphase promoting factor, is active, it is going to block securin. So if the securin is not active anymore, the separase will be free. And once it's free, it's going to cut the cohesin proteins so the chromosomes can separate. Okay, if we write it like a pathway, APC will be active. It is going to block the securin. And securin normally blocks separase. And separase normally cleaves the cohesins so that the anaphase will start. And this is the normal course of events. Now, once the anaphase starts, they are going to be separated. But actually, when the chromosomes are separating, two things are happening at the same time. So one of them is that the chromosomes are pulled with the help of this kinetochore apparatus. So because these microtubules, they are getting shorter, the kinetochore microtubules, they are getting shorter, they also have attached motor proteins on them, they are going to pull the chromosome. This is the first vector in this direction. This is vector V1, let's say. But there is another vector here. The cell used to be more like round previously. And then as the anaphase continues, it is going to elongate, which means it is going to be longer and thinner. It's because of these asters. Okay? The asters, they are attached to the plasma membrane and they are pulling the whole spindle apparatus. They are pulled by the asters. So previously, the spindle apparatus are here. So as it elongates, the spindle apparatus are going further and further away. So there is another vector, V2, here applied by these microtubules. So combination of these two forces, finally the chromosomes are separated successfully. And you know what happens if it is not separated successfully? Trisomy or monosomy, which unfortunately we see in cases, is also commonly seen in the cancer cells. Okay. Okay, the APC is a multimolecule. It is doing lots of things. The APC itself is controlled. If this connection, the kinetochore microtubule connection, is not done by all of the chromosomes, it is going to stop the APC. So if there is any kind of unattached molecules, this is going to stop APC from working. So until every single chromosome is attached to the kinetochore apparatus, the anaphase will not start, because you know that it's going to mean trisomy. Okay, this is how we control it. Another thing is that done or happening in this moment, not in this moment, but a little bit previously, is that the degradation of the nucleus. Nucleus is not actually completely degraded. It is fragmented. It is broken into vesicles and pieces by phosphorylating the nuclear lamina. The nuclear pore complex, the nuclear lamina, all together, they are broken into little pieces, and once the anaphase is over, once the telophase starts, it starts to come back and forms again the nuclear envelope, because the microtubules, they don't have to interact with the chromosomes anymore. You can freely reform the how to say, the nuclear envelopes. It's okay. And everything that we have talked about, all the cell cycle elements here, they're in a consecutive way. Before G1 finishes, S cannot start. Before S finishes, G2 cannot start. Before G2 finishes, M cannot start. Here, it's kind of more relaxed when we come to the cytokinesis. Cytokinesis actually starts before the mitosis finishes. So we can say that mitosis and cytokinesis actually overlaps. When the anaphase starts, actually cytokinesis also starts, because it starts here in the midline, which is the equator, which is determined with the help of the interpolar microtubules. And of course, it is not happening inside the cell, it is happening kind of outside the cell with the cleavage furrow, which is made of actin cytoskeleton plus myosin motor proteins. And this actin plus myosin motor proteins, which is altogether called contractile ring, is so strong it can actually break a glass. I mean, if there is a glass as thick as the cell, it can easily break it. Okay? It is happening outside it, and it is here breaking the cell into two. You can see it happening here. Okay, here there is the contractile ring, and you have to keep in mind that the cell morphology is also important. Normally, as you're going to see in the cell cycle and the cell culture class, the cells are spread, which means they are attaching on the surface. Here there is a substrate, and the cell is usually flat, and between the substrate and the cell there are lots of interactions, strong biochemical interactions. This fixes the cell over a surface, because to survive, to keep in a normal life, the cells, they have to be fixed themselves on a surface, otherwise they can do metastasis, which should be blocked. But you cannot cleave the cell in this time, because it's not possible. You cannot make sure about the cytoplasmic elements. You cannot make sure that you can divide it this way. So what is going to happen is that it will first cleave these attachments. So they're gonna be cleaved first enzymatically, and the cells, they're going to round up temporarily. You know that the M phase takes only 1 hour, and the first half is mostly prophase, prometaphase, metaphase, which can happen mostly here, but for the cytokinesis, they should round up. And next, once they are divided into two with the help of the cleavage furrow, they can now spread again. Where is Son Nicholas? And these establishments will be done back again, because without them, the cell will go under stress, attachment stress, and this is a useful stress. It prevents the metastasis. And when we come to the plant cells, of course, these things cannot happen. The plant cell cannot change shape. So what's going to happen is that we're just going to make a new cell wall in between with the help of some vesicles. These vesicles are carrying the material of the cell wall and plasma membrane. When they fuse, automatically they form this. Okay? And again, the location of this cell wall, the new cell wall, is determined by the interpolar microtubules, the equator. Okay, here we finished the cell cycle. I really hope I didn't forget something